Testing is based on the fact that we are going to study the effect of a single dose on a particular animal species. For two different animal species, usually we have one rodent and one non-rodent as the model, as per the Noel and EL to OAE at different dose levels, and the effect is observed for 14 days. All mortalities caused by the investigational products during the experimental period are recorded, and the morphological, biochemical, pathological, and histological changes in dead animals are investigated. That's the way how you go about doing that. So when we say the test report of this particular study, we would have these details being there. So what are these details which we are going to be having in that is, first you will have the test substance, the physical nature of the test substance, purity parameters, physicochemical parameters, identification data, source of the substance, batch numbers, etc. Okay, is there any question, beta? You guys are asking something? Uh, was it someone asking a question or something like that, beta? No? Okay. So in the acute toxicity studies, the signs which are recorded are increased motor activity, decreased motor activity, tremors and ataxia, tonic convulsions, catatonia, loss of writing reflex, sedation, muscle relaxation, piloerection, that is just called as strop tail phenomenon. You can see the center wala mice showing it out. So you can see the erected hair in that one. Cannibalism, you also say it as zombie effect. Strobe reaction, hypnosis, analgesia, lacrimation, diarrhea, salivation, which can be viscid or watery depending on what the type of salivation is exhibited, breathing, that's just um, extensive vibrational phenomenon seen in the entire body, respiration, it can also lead to depression, stimulation, or failure, skin color changes. So these are the ones which you would be looking at acute toxicity levels in the acute toxicity studies. So what are we going to be calculating? So approximate lethal dose method. Here approximate lethal dose method is sequential dosing until lowest lethal dose is obtained. The lethal dose thus obtained is approximate lethal dose for 6 of the 10 animals are required to achieve approximate lethal dose. So the range which is being there is that you may have for statistical significance at least 6 to 10 animals being there. The class method is class method diluted or undiluted material. Test material is given to several groups. A vehicle control group is included which checks for clinical signs, morbidity and mortality. Fixed dose procedure is a method in which the dosing of animal is a stepwise fashion using fixed dose of 50, 500, 200 mg kg per kg body weight. Traditional testing limit is normally 50 milligram per kg weight of the part. Observation, what you look out is for mortality, death rate or death time point, time course of signs of toxicity, and necropsy finding. That means post-death, the autopsy finding details. So when we look at the acute toxic class method, the acute toxic class method is a fixed dose level. It's 550, 300 or 2000 mg per kg. The initial dose is related based on the assumption that using minimum number of animal in stepwise procedure will provide enough information on the acute toxicity of the substance. Up and down procedures, up and down procedures or animals are dosed at one time, minimum of 48 hours interval. First animal survives, next one receives higher dose. The spacing of dose is adjusted up or down by a factor of 3.2. So here we are trying to find out acute toxicity at what concentration levels by giving a single sharp dose and checking out for its survivability. So next is the one where you look into repeated dose studies, which are called as subacute or chronic toxicity. So when you look into subacute or chronic toxicity, you would preferably use a non-rodent. So normally the non-rodents are to mammalian species, okay, basically because you would want it to have similarity in terms of structure and physiology with humans. Dose is independent on dose escalating studies. Drug administered is by clinical route. You check out or you study the behavioral pattern, physiological conditioning, and biochemical and microscopic observations. So in the biochemical observations, you'll be understanding the ADME concepts in detail. In microscopic observation, you are trying to understand the way how it is going to be settling down in individual cells. For example, those which have a tendency for certain tissues tend to get accumulated in that particular tissue, so that is being checked out. And basically, rodents are non-rodent species. Dose is expected therapeutic level daily or expected therapeutic level to increase in a phased manner, not a direct way, phased manner. That means you will increase it slowly, proportionately in the phased manner. The test substance is usually given oral about equal to greater than 90 days observation point of view. You would have a regular body weight variation checked out, biochemical parameters checked out, cardiovascular parameters checked out, behavioral changes observed out. At the end of the study, experimental animals are sacrificed and the gross pathological changes are observed and all tissues are subjected to histopathological analysis. There should be little individual variation between animals and allowed weight variation range is not more than 20%, plus minus 10 to 20%, of which 20 is the upper limit. It is used to determine the maximum tolerable dose, and that's what is the major intention. So here the test compounds are administered on a 14 days via the route of administration, okay, intraperitoneal, intramuscular, paraoral, or subcutaneous for the route of intended for human administration. Animals are observed individually after dosing, periodically during 24 hours, and thereafter for 14 days. The long-term study which gives you an idea about the nature of toxicity. So the types of local toxicity studies which we entail into which we study are dermal toxicity studies, that means skin-based toxicity studies. Dermal phototoxicity studies, where you would want to know the chemical compounds, has it make the skin sensitive to phototoxic effects. Vaginal toxicity studies, rectal tolerance studies, these are the ones which are called as local toxicity studies. So the parameters monitored in all these studies are examination of erythema and edema formation, observation of swelling, histopathology of vaginal wall in terms of vaginal toxicity studies, signs of pains, blood or mucus in all of them, histological examination of rectal mucosa in rectal tolerance studies. Is this clear till here, beta? Do you have any questions pertaining to it? Any
No, sir.
questions, guys? Okay, it's clear. All right. Thank you so much, beta.
So can you explain about phototoxicity? What does
Sorry, beta.
phototoxicity studies? What does it mean?
Phototoxicity, beta. You want to know about phototoxicity? Okay, beta.
Yes.
Okay, beta. Light-based sensitization, beta. Do you know something called smog? Have you heard the word smog, beta?
Yes, sir. Yes, sir.
It's smoke plus fog. One of the common prevalent things which occurs in polluted region. For example, if you take Delhi as an example, Delhi is our capital. At the same time, it has a higher pollution level in the winter season because of smog effect. Because it's right now cold there, cold condition. Okay, so there is a fog, and if the smoke content is higher, it becomes smog. Now, smog is possibly more disastrous because it has photochemical reactivity. Photochemical reactivity in the sense it will show chemical reaction light. If I have to, uh, do you know that certain solutions or certain compounds are kept in amber colored bottles in the lab, right? Do you know
Yes.
any of them which you can tell me which is
Acetone?
kept in? Okay, right, that's good. Another one, for example, is silver nitrate. Okay, it's also kept in, the liquid is kept in amber colored bottles. The reason, you know why? Because if it is kept in transparent one, it will react. So you call it as photochemical reaction effects, beta. Now, normally our skins are non-reactive to photochemical action. That means we, when exposed to sunlight, may feel the heat, okay, may feel the normal no intense heat in entity, warming up phenomenon, sweating, these are the responses which you'll get, okay, but you would not have a reactivity. So albeit reactivity is shown by which particular part. So for example, you have known a disease called albinism. Do you know about albinism? जिसमें बेटा ऊपर
Yes,
का
sir.
स्किन निकल जाता है, फेयर हो जाता है, वाइटिश वाला स्किन दिखना शुरू हो जाता है, राइट बेटा? तो वो पर्सन अगर कभी सनलाइट में चले, दोज हु आर हैविंग एल्बिनिज्म, तो आप देखोगे 1 मिनट में ही उनका पूरा का पूरा स्किन रेड हो जाता है. अगर उनको सनलाइट have minor bleeding from the small pores outside, and it will become uh, instrumentally infectious for them. That is because the skin pigmentation doesn't have melanin formation, doesn't have keratin to higher content, and lack of these two entities, these two proteins which are protective in nature, make that skin photoreactive. Okay, beta. So if your chemical substance which you have taken affects your melanin pigment formation, melanin pigment formation occurs in the presence of sunlight, vitamin D conversion. Okay, or if it affects your keratin levels, these are both proteins. Okay, by either chelation or by any of the chemical reactivity, then you call it as phototoxic effects, beta. Okay, so phototoxic effects are seen at the dermal layer. तो कोई कंपाउंड जिसके खाने से आपके स्किन सरफेस पे रैशेस के बजाय में फोटो रिएक्टिव इफेक्ट्स प्रोड्यूस आउट हो तो उसे आप बोलोगे फोटोटॉक्सिसिटी. इज इट क्लियर नाउ बेटा?
Yes, sir.
Okay बेटा, थैंक यू सो मच. So let's continue. But you will hear this word OECD guidelines. Okay, now this is the guidelines which have been made, which state that to be done for every substance which you can think is coming in contact with skin of human beings. So chemical preparations, pesticides, industrial chemicals, everything needs to have this tested being done. For example, the section 1 says physical chemical properties, second says effect on biotic systems, section 3 says degradation and accumulation rates, then 4 says health effect, specifically which you can use or check out for by software which is mentioned out here, software TG455, TG432, TG425, and the earlier for biotic system was TG223. The other test guidelines, list of adopted test guidelines, including dates of revision, list of the toxicity guidelines and addenda adopted by the council. So these are the ones which you'll be looking out into. So TG here starts for the software for example, TG 455 is a toxicity guideline 455, which has been identified by the software systems and the systems have been made aligned to detect it in the same range. So guys, you can ponder over this part. Parameters measured in acute toxicity: LD50, LD50, LC50, LD0, LD10, EDs and TIs. Just go through it, beta. If you have any doubts, I'll just wait for a minute or two and then I can explain. If otherwise, we can move on. Is it clear, guys? Do you have any doubts? Anything you want me to explain on this?
Sir, yes, sir. Sir, how is the dose different from the concentration, LD50 and LC50? Because
Acha, acha,
eventually
acha.
it's the
Okay, okay,
same
okay,
thing.
okay.
It seems
Okay,
like at
beta.
least.
Okay, 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 okay. Now, beta, look at both of them, beta. For example, when you say lethal dose, okay, and lethal concentration, beta. These are two parameters which I was trying to be doing. For example, there are neurotoxic agents. Let me give an example which you can understand very. Okay, chloroform, beta. Now, chloroform was earlier used to make a person unconscious. Okay, but बाद में उसको बंद कर दिया गया. Do you know the reason, बेटा, क्यों क्लोरोफॉर्म क्यों बंद किया गया था? Why is now chloroform not used for making someone unconscious, something like that? Any idea,
Yes.
guys? Because beta, it gets converted into a poisonous gas called phosgene. ओके एंड फॉस्जीन किल्स, यानी आपने क्लोरोफॉर्म एक्सपोजर में आपने बेहोश करने के लिए किया रहेगा, अगर लेकिन उसका एक डोज लेवल एक लेवल से ऊपर चला जाए तो इट इज फॉस्जीन विच इज गोना किल आउट. ओके, नाउ व्हाई एम आई टेलिंग दिस थिंग? जब हम लोग डोज की बात कर रहे हैं तो इट इज व्हाट वी आर गिविंग टू द एनिमल. ओके, नाउ दैट वी आर श्योर अबाउट based on which sort of which sort of dosing we have done. Like for example, if we have done as peritoneal, how much is going? If you have done it intravenous, how much is going? We are very sure about it. Okay, so in dose, what we are doing is we are making that chemical substance reach the plasma. That means inside the blood. So that is attributed as lethal dose levels. So that is LD50. So 50% of the mortality causing dose will be called as lethal dose 50. But in another cases, for example, like gases, for example, carbon dioxide, carbon monoxide in certain level is detrimental. It will absolutely kill you. Okay, first it will take you to coma and then kill you because it gets attached with the hemoglobin, forming carboxyhemoglobin, which is an irreversible reaction. So here you are not deciding the dose. The dose decision is by the way the respiratory system is taking in how much. Okay, so there we only know what is the concentration of the gas or concentration of the liquid where this organism is going into. Okay, in that sort of a case we will call it as L50. So here in toxicological studies, it is how much gas or how much liquid has been taken in by the organism when the person is in a situation. So here it can be, if it is in a gas chamber, how much gas was inhaled by a person which caused the death of that person will be called as LC50. Because here we are not intertwining, we are not pushing the material into the blood plasma, it is being inhaled in. That's the time when we call it as LC. Is it clear?
Yes, sir. So basically, LD50 is something that we can control, and
Yeah,
LC50 is not.
LD50 beta we can control, beta LC we can't. For example, if somebody say somebody is very good at holding the breath. Okay, say a person is able to hold the breath for say the world record is 22 minutes underwater. Okay, so let's say somebody holds breath for say about 7 minutes odd. Okay, and you put two people into the gas chamber, just like gas chamber which was existent in the German time point World War. Okay, so they were using neurotoxic gases and they exposed this thing to 5 minutes, assuming that both of them will be dead. So what happens is after 5 minutes, you had put two-person into the gas chamber and you had exposed it for 5 minutes. So what happened? Within the first few minutes, the person who did not have the capability to hold the breath is already dead. But after 5 minutes, this guy was able to hold the breath, and then you would see the chamber door opening just to throw the body out or stuff like that. So what you saw was in one of the gas chambers, you could see that the person was carried out and he acted dead, but he is alive because it was his capability to hold the breath for a certain duration. Similarly, the respiratory rate of every animal is different. Respiratory rate for humans are also different. person to person is also different. So there is a variation beta. So here you can say ki what is the concentration at which the inhalation occurs and the death occurs. Hence you say LC50 beta. Okay?
Yes, sir. Understood.
Let's continue now. So, beta, this is the comparison which is being done and observed parameters being measured for all these three cases which you are aware. So, rats preferred for oral and inhalation tests, rabbits preferred for dermal tests. Rodents, usually rats preferred for oral and inhalation studies, rabbits for dermal studies because rabbits are very strong in that part. And non-rodents, usually dogs, recommended as a second species for oral test because it's fine. But if you can't have the dogs for inhalation studies because it's very difficult. They are the most sensitive guys in terms of nasal inhalation parameters, so they may not be very conducive and compliant to it. So number of animals you are seeing 5, 10, and 20. Okay, so that's an averaging. Young adults is what preferred out. Dosages are 3 dose levels, which is normally nosed out. Observation period is 14, 90, and 24 months. So 49, 90 are days, and 24 months is about approximately 2 years. That's what normal time point is. So when we look into parameters, LD50 is for the 14-day first ones, while 90-day for behaviorals and 20-24 months for the behavior, physical, microscopic, histological, and necropsy studies. So this was the one which is toxicity studies beta, methods of testing and details. Hope this is clear to you. What I'll also do is I'll also send across the video to Payal and Shruti ma'am. So you can go through it whenever you wish to. It's a 7-minute odd detail, so it will give you an idea about toxicity testing. Hope, is it fine, beta? Meanwhile, any questions? Hello guys, any questions, beta? Okay.
No, sir.
Okay, thanks, beta. So next, what I'll do is I'll have something of toxicology, just a general version of 8 point, about 4 minutes detail of toxicology, which I'll show it to you. This is a comparison of acute versus chronic toxicity. So guys, this is very clear to you. Any questions on chronic and acute toxicity now, beta? Hello guys, you around, beta?
No,
Any
sir.
questions? Any questions? No. Is it
No, sir.
clear? Okay, beta. Thank you.
Yes, sir.
So I'll play one more, beta, one last one. There's a long video. So this is about toxicology. It's a little bit long video, okay, of 4 minutes.
Excuse me. Hello, sir.
Yes, beta.
there an audio to this video, or is it just a video?
Uh, you're not able to hear the audio or video, both, or
Not able to hear. We can see the video.
Audible.
okay, 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 okay. One second, beta. I'll just close and then open it up. I'll just see what's wrong with it. No problem. Thanks, beta. Thanks for telling me. Okay, this seems to be. Can you hear it now or see it?
So the video is visible, but the audio isn't visible.
Now, is it audible also, beta?
No, sir.
It's not audible.
No, sir.
Oh, one sec, guys. I'll just share again. Okay, guys. Can you see the screen now?
Yes, sir.
Okay, let me play, beta. Just see if you can hear. Now you can hear?
No, sir.
Oh,
Did you select the include computer audio option while screen sharing?
yes, beta. I had included because you could see the audio previous videos, na, beta. Right now, previous videos you could see, you could hear it also, na, beta.
No, sir.
No, sir.
Oh, something is wrong. Let me, beta. More action. Mm. Okay, but I'm playing once more. Just see if you can hear and tell me about. Okay, beta. One sec.
So, you want to know more about toxicology? Well,
Yes,
toxicology is also called the
Is it now coming, beta?
yes, sir, it's
Yes, sir.
audible.
It's audible.
Right? Okay, fine.
Yes.
science of poisons, is the study of the harmful effects of substances to living things. We normally think of poisons as stuff that can kill or seriously harm us, like cyanide or snake venom. But toxicology looks at the adverse effects of all substances. 500 years ago, the German-Swiss physician and alchemist Paracelsus, the father of toxicology, declared that only the dose is the. He realized everything could be toxic. Medicines and even essential chemicals like oxygen and water are harmful if you have too much. We often think only man-made chemicals are harmful, while natural chemicals are not. In reality, some of the most well-known poisons occur naturally, like strychnine from a tree of the same name, and botulinum toxin, deadly in food but widely used as the cosmetic treatment Botox. Chemicals make up everything around us: air, food, water, clothes, and mobile phones. A cup of coffee contains more than 1,000 different chemicals. You, me, and all living things are highly complex and organized collections of chemicals. These chemicals form cells, tissues, and organs, like the heart or lungs. Chemical reactions are continually taking place in our bodies and keep us alive. Our bodies react in many ways to excessive chemical exposure. Contact with skin might cause irritation or an allergic reaction, but most chemicals affect us when they are absorbed or injected. Each substance's ability to pass through the lungs, stomach lining, or skin depends on its properties, such as molecular size or water solubility. Once inside our bodies, chemicals can cause effects ranging from tiny, non-harmful changes to cells to sudden death. Sometimes ill health is not seen until years later, or even in future generations. Toxicologists identify the harmful effects caused by different chemicals and how much of the chemical is required to cause the effect. By doing this, they are able to provide advice on safe levels of exposure. In doing so, they must consider the variability in a chemical's effects between different species, males and females, children and adults, and healthy people versus those who are ill. Toxicology is a complex and diverse science. It has to be to discover the ways that chemicals are absorbed into, move around, and leave our bodies, and how they impact our biochemistry. Consequently, toxicology includes many specialisms, such as genetic toxicology, which looks at interactions with DNA, and reproductive toxicology, which studies potential impacts on fertility and the development of offspring. Although human safety is the original concern of toxicology, ecotoxicology is the equally important study of how chemicals affect wildlife and the ecology of our environment. Ultimately, we know there's no such thing as a safe chemical, but we also know there is no chemical that cannot be used safely by simply limiting the exposure. Toxicology is a fascinating and diverse area of study and work, and our collection of videos covers its many facets. Click on our links to see toxicologists talking about their specialist areas in more detail.
So guys, you got an idea about the toxicology. You saw some details also. I'll show you one more, which is a detail about it. Okay, so this is gonna be environmental toxicology. Okay, I'm gonna share this video with you, Bida.
Okay.
Okay.
We will be talking today about toxicology and
So this is gonna be pertaining to environmental toxicology. Just confirm, Bida, if you can see the screen and the slide.
Yes, sir. Yes, sir.
Okay, so I'm playing it out. Just checking the sound levels. It is at 72. Should I increase it out or is it okay?
Okay, sir. It is okay, sir.
Okay, I just increase a little bit beyond up to 80 only. That's not more than that, so that it doesn't rhyme high on the. Okay, playing it for you now.
its relevance in the medical field. I will be covering a few concepts that are highly mentioned in this field. And we will start by defining what is toxicology as a field, its different classes or categories, including, of course, environmental toxicology.
So you can hear it, Bida, clearly?
Yes, sir. Yes, sir.
Okay, okay, Bida, thank you so much. But this is the one which I am running this particular video based for one reason. This is about environmental toxicology, which is also part of the toxicology as per regulatory toxicology requirements. So this will cover up that part of it, and then I can go ahead with the clinical part of toxicology. Okay, guys. So playing it for you.
We will also learn and discuss about the dose-response relationship and how it relates to toxicity, the ADMET process, which is how chemical substances interact with human body, and last but not least, the applicability of toxicology in risk assessment and environmental regulations. Most of you are joining this webinar because of your academic background or your work area. However, toxicity is a common term in our life. For example, when you go to the food market, we can be concerned about where are our vegetables coming from, especially since sometimes vegetables or fish could be contaminated with metals, pesticides. Also, in our homes, we have detergents and paints and other substances that we might consider contaminants or irritants. So we, in a daily basis, are at some point concerned with toxicity. More recently, we are also highly concerned with the presence of plastic and constituents of plastic, such as phthalates, on toys, and the effect that they can have on health. In this case, also very recent is the interest of identifying what containers are appropriate to be used in the kitchen, especially if you want to use them for microwave heating. So as you can see, toxicology is not only important to those that work in the field, but really important to everybody. So having that in mind, what is toxicology? Toxicology is the science that focuses on the adverse effects or toxicity of chemical and living organisms. It's been called the science of poisons. Also, you will be listening some concepts today that might relate to pharmacology, and it does relate to pharmacology. But in this case, pharmacology focuses on the potential therapeutic or beneficial applications. So if you want to compare toxicity or toxicology with pharmacology, the simplest way would be to say toxicology is concerned with the adverse effect, but pharmacology is mainly with therapeutic effects. However, toxicology is a wide field, and you would hear different type of toxicology. For example, forensic toxicology. Forensic toxicology is concerned with medical legal aspects of the effects of the drugs on humans or in animals. It's really a hybrid with analytical chemistry and toxicology because of the methods that are used. And the purpose is to establish the cause of death and circumstances in postmortem investigations. Also, a very close science, the clinical toxicology, that sometimes is confused. In this case, clinical toxicology is concerned with diseases caused by toxic substances, and it's mainly applied by physicians in emergency medicine and poison management. And the main contribution of clinical toxicology is to determine or to provide with new disintoxication treatments or techniques. Another highly applied area of toxicology is molecular toxicology. It's concerned with the cellular events that occur at the cellular level or biochemical interactions as a consequence of the exposure to certain substances. It's not only important in environmental toxicology or in environmental science, it's highly important and necessary in drug development. And those of you that are interested in natural products, keep in mind that just because they come from nature does not mean they cannot have some effects. And we hope that they're all beneficial effects. But in order to prove that, we need to have molecular toxicology implemented to define at what level or what kind of interaction those substances are having with DNA, with lipids, with proteins, and so on. Similarly, in the environmental area, the insecticide development area will need that kind of studies. So we know that insecticides will not affect equally every species, but we need to know exactly what is the mechanism of action involved in that effect. Sadly, when scientists don't take the time, or maybe pharmacies don't take the time to develop the appropriate drug testing studies, such as molecular studies, will result in effects in the long term. For example, a typical example that you could read is the case of thalidomide. Thalidomide was a sleeping aid medication recommended during the 50s as a very secure and safe medication. So secure, they thought at the time, that even pregnant women or children could ingest. sadly to say, they lack of studies indicating that if thalidomide had or not the potential to cross the placental barrier. And that is a key study that needs to be done in order to recommend any women to ingest any medication. The purpose actually for pregnant women to use it was to ease the first trimester secondary effects, nausea. It will relax the women at the same time as it will be a sleeping aid. Unfortunately, long-term was evidence to show that children, offspring of these women, would be born with specific birth defects, especially it would affect the development of the limbs or extremities such as arms. And you can see them, this is some of the images of those children. It was also understood later that the reason for that effect was that it was interfere with formation of blood vessels. Currently, it's still used for other purposes. Of course, it was taken off of the market, or then it came back. And nowadays, it's used for cancer and leprosy as well. Another area of toxicology is regulatory toxicology, very important for environmental field and establishing risk assessment. It's really responsible for deciding whether a substance possesses low health risk. So you could say regulatory toxicology is really putting all together levels that should be safe in order to make safe recommendations.
So beta, this is the regulatory toxicology which we are talking about. So this comes under this overall categorization of environmental toxicology. So it's good to know the overall perspective, and then we go to the specifics of this particular subgroup of regulatory toxicology beta. So that was the reason I thought I'll run through this particular presentation from Beatrice Ayers, who is very good at telling about environmental toxicology details. So let's go through it, and then what will happen is we will understand the entire perspective, and then I can go ahead with only the clinical part of it, which is regulatory toxicology, which is remaining for you. Kimra?
All of these agencies, regulatory agencies, rely on regulatory toxicology in order, for example, for FDA to determine what can be sold, for EPA to determine what are the acceptable levels of chemicals in the environment. OSHA also relies on it, and DOT, Department of Transportation, as well. Now, getting a little more into the subject for today, which is environmental toxicology. Environmental toxicology focuses on the impact of pollution in the environment, and specifically on biological organisms, both human and non-human. Of course, we want to protect human, but we cannot do only studies with human. We would try not to, unless it's an occupational setting. So toxicologists rely a lot on other models such as using fish, birds, and other terrestrial animals. Within the environmental toxicology, you would have a subdivision, which is called ecotoxicology, which mainly focuses on the effects of pollutants on the population dynamic within an ecosystem. So it's still interested on the individual, but also on what happens when those pollutants are in an ecosystem and how it interferes with the dynamic. So for you that are in the public health area or biology or chemistry, you could clearly see how environmental toxicology is a multidisciplinary science where multiple areas are integrated. And I think that's the beauty of it, is that it's not only a pure science, it's an applied science. So one of the questions that I hear continuously is, why would we see that even the same substance would affect certain people, but not everybody? And a typical question will be in the case of cigarette smoke. Well, there's multiple factors. It's not only about the substance. There's also other factors that affect toxicity. In this case, we have the period or time of exposure. How long have we been exposed to that substance? The source, how are we exposed? The route, how does it gets into the body or into the subject? Subject susceptibilities. We all know that we're not the same. There's variability among subjects, and also there's certain conditions or diseases that might make a person more susceptible than others. Other age, It's age dependent. Also, there are factors that have to do with the age. So that will be another factor. The concentration or the dose is definitely a key aspect, and we'll talk more about dose as the presentation continues. Chemical substances and chemical interactions. In the case of the properties, chemical and physical properties of the substances will determine if they can be really absorbed into the body or not. And also the chemical interactions. We are not only exposed to one or two substances, most of the time we're exposed to multiple substances, and that will determine also the toxicity. Going a little bit over those, when we talk about the period or time of exposure, there's three main ranges: acute toxicity, subchronic toxicity, and chronic toxicity. Acute really relates to a single or a brief period of time. Subchronic will be continuous or repeated exposure to months. But chronic, it's in general, is considered as a very long period of time or years or lifetime. These are kind of relative concepts, but they have to be well defined in any studies. So when you go to literature, be sure that the toxicity is also defined by a period or time. Besides how long are we exposed, also how are we exposed? And we can be exposed to pollutants through air, to water, soil, crops, food. And together with exposure is a concept known as fate and transport. As you can see, we can be exposed to different sources at the same time. These illustrations is just simplification. We could be exposed to surface water, to contact in air, also groundwater. And as you can see, chemicals will behave differently in different compartments. So they will not behave the same in water as they do in air or in soil. And that is very important. Also keep in mind that once they are in the environment, they will encounter situations that will transform them. So they might not all be available even when they are present in the environment. They might not be available to be uptaken. Another key important factor or characteristic is how lipophilic or volatile these substances are. If they are very volatile, they'll go more into air. If they're lipophilic, they might interact with organisms. There's many contaminants that we might be interested. However, I'm going to mention this, which are considered main or criteria air pollutants. Criteria air pollutants are contaminants that are found all over the United States. Other places in the globe as well, but our interest here and the way we regulate this is through US, and can harm health, it can harm human, it could affect the environment, and also cause property damage. So these are the main pollutants: sulfur oxide, nitric oxide, carbon monoxide, ozone, particulate matter in different sizes, metals, and volatile organic compounds. When we talk about how these substances move to air in this case, we should consider that air is the primary route of contaminant through evaporation, stack emissions, and air release. However, then since you should consider that they might be less viscosity in air, contaminants move faster in the atmosphere than in water. That could vary from place to place, since there are some places have higher humidity. Still, the viscosity will be less than in water. However, besides that, there's other variables that will interfere on how these contaminants are diffused through the air, such as molecular weight compared to the air, the temperature, the energy of the molecular interactions, the wind currents, and of course, the heat transfer from ground. So perhaps, closer to Earth, you will have the effects of what's happening in that area. If we're thinking about water, how contaminants will move in water, they enter water by direct applications, through spills, wet and dry deposition, and it also interface movement within levels of the water, and also it will be affected by the proximity to soil. So if you have water that is closer to land, you will have contact with land, and that will definitely affect how these substances move in water. Also, other variables that affect how pollutants move in water will be the molecular weight of this pollutant compared to water, the water temperature, and of course, how soluble this substance is in water, and the concentration. Also, keep in mind, based on the image, that some of these substances might be hydrophilic and or lipophilic, and they might be absorbed by some of the species in water as well. Soil is also a way we can be exposed, and in this case, the characteristics of the soil, the porosity of the soil can vary with composition. For example, the percentage of salt, sand, silt, clay, organic material, all of that will affect how these pollutants move in sediments or in soils. So the transport of the contaminant will depend also on the particle size of the soil. And the humidity, keep in mind that also how humid or dry a land can be will affect how it moves. The molecular weight of the pollutant or contaminant, and how long these contaminants are in sediment, and the concentration. But something particular from soil in comparison with water and air is that in soil, we would have also biodegradation of the contaminants, or decomposition. And that can happen in air or in water, but very minimum. More in soil, maybe water, air, since they don't stay in the same place, they move from one place to another. You will mainly see this kind of effect, biodegradation, which is the composition by microorganisms in soil. And all of that is important, because I guess that the key question here is, okay, a substance can be in air and water or in soil. Does that mean the organism will uptake that contaminant? Well, actually, the good news is that not always, and not at the same amount. In the environment, only a portion of the contaminant is potentially uptaked by the organism. First, they could be transformations in the environment. For example, the amount of mercury in sediment. In sediment, you'll have microorganisms that will degrade or biotransform the mercury, and they might not be available to be intaked by an organism. So it might be there, it might not be as easy for an organism to uptake it. The same happens with organic chemicals in soil that will be decomposed or biodegraded by other microorganisms, and might not be as available to be uptaken by us or other species. A key aspect also of how toxicity happens are chemical interactions. We all know that we're not only exposed to one substance, we are exposed to multiple substances.
So beta, here the concept of LC50 comes. So when there is exposure, for example, the exposure, the quantum of exposure is based on concentration, because we are not dosing per se, is the exposure per se. So what I'm trying to tell you is, for example, if you take I'll give you a very clinical example. Let's say there are two smokers. Okay. Now one smoker lives in Delhi and the other person lives in, say, Himachal Pradesh, somewhere in Himachal Pradesh, Shimla or something like that. Do you think the LC50 or the concentration ranges of chemical, say nicotine or say tar, in the body of both the persons would be same or will be different? Yes, guys? Would it be same or would it be different, beta?
Different.
Hello?
Sir, it will be different.
It will be different. Why? Because beta, in Delhi, because of the existing pollution level only, a normal person would be showing a susceptibility range which is higher because of the environmental reason. So what happens is this becomes a serious problem when we are conducting, say, a BAB studies or a clinical trial. Okay, because in a clinical trial or a BAB study, you may have people from the world, so MNC based ones. So if you recruit, say, patients from Delhi, okay, you have patients from Shanghai in China, and you have patients from, say, London in Europe, Mumbai, Calcutta, and stuff like that. And then you pull up the data and try to deduce out some information. So what will happen is, because of the city level pollution levels, which is environmental toxicology effects, the parameters studied will tilt or would be skewed towards one side because of external environmental factors which we are not considering. Hence, in rec toxicology, it is also supposed to be known that if you are having comparison being made, it has to be, the subjects have to be in the same location, same zones also. So just to ensure that the environmental conditions which they undergo are nearly same. So this is also well looked into when there is an occupational hazard point of view, which is also checked out. For example, silicosis, asbestosis. You have heard of these words, beta? Silicosis, asbestosis. Anyone, guys? You have heard of it, beta. So one is silica dust. So silica dust is people who are working with tile work, granaries, and stuff like that. They have small particles accumulate in the lungs. Asbestos is somebody who is working with the asbestos sheet industry. They will have asbestosis fibers settling down in the lungs. Now, added on to it, you have some exposure of mnemonic phenomenon being studied. So what will happen is, predisposition to this occupational hazards, okay, of environmental factors would also get added to it. And if you do not consider that, then what the problem would be is that you would find that your drug is ineffective or not showing an effect as anticipated, okay, because you have not looked into the parameters which are about environmental toxicology parameters too. So that's what is being tried to be explained out here, beta. That's the reason why we say LC50 and LD50 are different, beta. Okay, hope this is very clear, beta. Please say yes. Thank you, beta. Thanks so much. So I'm playing it further now, guys.
Unless it's a very well-controlled occupational setting where you can say, well, this is the only substance that we handle here. Most of the time, we're exposed to multiple substances. As well happens when a patient ingests more than one drug. And also at home, we use products for our care or skin that could also interfere with the toxicity of these substances. But when we talk about chemical interactions, there are specific ones. Here in the environmental field, there are two main ones: antagonistic effects and synergistic effects. So one pollutant can affect the toxicity of the other. When we talk about antagonistic effects, we refer to decreased toxicity. So many times we are exposed to certain contaminants or lifestyles, and we are advised to ingest more vitamin C or certain products from plants that might decrease or minimize the effects. When we talk about synergistic effects, we talk about an increase in toxicity of some chemicals. A typical example of that would be the interaction between asbestos and cigarette smoke. If an employee works with asbestos, or if you work in an area where is an old building with still asbestos in their walls, and you also smoke, there's an increase in the chances of developing lung cancer. So a synergistic effect is clear between asbestos and cigarette, since it can increase the incidence of lung cancer. So we've been talking about effects. So, and that's kind of a generic term. There's different type of effects, and for that, perhaps in another webinar, we can talk about them. But I'm going to classify the effects in two groups: specific toxic effects, which are directed to a target tissue. It's a toxicity that to a particular tissue or organ. And these tissues are called target tissues or target organs. But there's also systemic toxic effect, which is a more general effect. Remember, us as a organ system, we are composed by organs, by tissues, and by cells. And when we talk about systemic toxicity, you could identify toxicity at all of those levels. However, those is at what level will we really see those effects? In order to determine at what level, we need to define what is dose. And for that is this concept of dose-response relationship. Dose, as defined by Paracelsus, he would say that only the dose makes the poison. In other words, any chemical is capable of evoking an effect. And we can all relate to that with a simple example as caffeine. For some people, they could take 1 cup of caffeine in the morning and they will feel fine, but if they take 2 or 3 during the day, they will feel agitated or nervous. So that will be a simple example that the dose really can make the effect. But in terms of definition, dose is the amount of the material needed to produce an adverse effect. And response is that adverse effect that we are seeing associated to that substance. That would be in toxicology, because in pharmacology, dose is the amount needed to produce a good effect, a therapeutic effect. And response is the adverse effect, the beneficial effect. Of course, that would be in general, because if we were talking about chemotherapy, the response would also be an adverse effect, killing malignant cells. However, for those of you that don't work directly with these assays, if you are relying on literature, I would urge you to go and always look at the design of the study. And if you really want to be certain or say that a substance induced an effect, be sure how the study was taking place and what are the treated subjects and what are the controls. Controls are that group, that population, it could be animals, could be humans, that have the similar characteristics than those treated, but that have not been treated. Therefore, any effects that you are monitoring can only be associated or mainly be associated to that substance or to that dose. However, how can we translate those concepts into numbers, into levels to be used for regulation? In this case, we use the dose response curve. The purpose of this curve is to illustrate how the response changes as a function of dose. As you can see, the graph in the x-axis, you have the log dose. In this case, you can represent it as log, but it doesn't have to be logarithmic. It could be also micromolar. It could be milligram per kilogram of body weight. It all depends on your analysis. And the response, it will be, what are you looking for? What is the effect that you're looking for? Most of the time, we define dose response with lethal doses. What dose we're going to see lethality or death, growth inhibition. That happens a lot when you're working with cell culture. But if you're measuring other effects, you could call them effective concentration. An example will be if you're identifying some kind of substance that helps hair growth. So the effective concentration will be at what dose you will start to see hair to grow. So you could use any of these and represent it in a dose response curve. Let's go more specifically. Let's give you some examples. In this case, we have the dose response curve for non-carcinogens. You will have here 3 endpoints: liver damage, developmental toxicity, and mortality. And you have, in this case, the dose is represented by milligram per kilogram of body weight. And that's, you can understand, depends the size of the subject. In this case, are not subjects, they're animals, but dependence on the size of the animal is the dose, the milligram per kilogram per body weight. And the response is represented in percentage. So let's go and see if you look at the curve at 100 milligram per kilogram of body weight, you will not see any effect. No liver damage, or at least not that they could identify at the moment. No liver damage, no developmental toxicity, and no mortality at 100. So that will be kind of the safe dose. So as we can move to 1,000 milligram, you will see that at that dose, you have 10% of the population have already represented liver damage, have shown liver damage. But if we move to the right, at 10,000 milligram, you will see mortality. Not only mortality, you will see the LD50. That will be more lethal dose in 50% of the population. You could also read LD25, LD90. So that only represents the percentage of the population that has expressed that damage. Same thing will be with IC50. It will be the 50% that inhibits the growth of the population. And going back to the 100 milligram per kilogram, which might seem like the safest concentration to recommend, definitely that would be the homework to take from that curve. Because what we want to identify really is the NOEL, the no observed effect level. We want to identify a concentration that where we don't see any effect. And for that, that will be the NOEL. Also, there's other ways of expressing doses. You could see the LOL, which is the lowest observed effect level, is the concentration, the lowest concentration where you will see an effect, but this is the lowest. And the same thing would happen for adverse. So we might be looking for a concentration where we can be sure that there's no adverse effect. And adverse effect always refers to lethality. However, you might say, well, then
So guys, is this clear for the way how it is explained for Noel? Any questions pertaining to that for me?
No, sir.
No questions. Okay, fine. Take it. Thank you.
I can feel comfortable with using 100 milligram per kilogram. Well, no, not exactly. That concentration is used to generate a reference dose, and that's the reference dose that is recommended for human. And why is that? Is because most of the NOEL are obtained from animal studies, and we might not react similarly to animals. So therefore, we need to take into consideration other variables, other factors that might interfere. There's interspecies variability, but there's also intraspecies variability. And keeping that in mind, in the 1950s, two researchers from the FDA introduced a concept called safety factors or uncertainty factors. And what they do, or what they recommended at that time and has been implemented since, is to, once we have a NOEL, a no observed effect level dose from an animal study, we want to make it a reference dose for human. We should integrate other factors and make it a smaller concentration or smaller dose. So all the purpose here is to extrapolate from animal studies to human studies using the lower concentration possible in order to assure the safety of human. Here will be a comparison of how to apply a reference dose concept. Here we have aldrin and DDT, both have liver damage. However, if you compare at the end of the table, what you will see is that ethylene glycol with 2 milligram will pretty much have the same, it's a reference dose for that substance, while for aldrin and DDT is much, much lower. That clearly is an indication of the toxicity and how potent a substance could be in comparison to another. But if you want to hear about something a little bit more related to you, here is a summary of common substances and those doses that are non-toxic versus toxic doses. So you would have, in the case of alcohol, they have studies have indicated with 0.05 is a non-toxic dose. I shouldn't say beneficial, but I could say non-toxic. However, 10 times more with a 0.5%, it will result in lethality. So that's a lethal dose. The same thing would happen with carbon monoxide that is present in cigarette smoke. Less than 10% is considered a non-toxic, but over 60% is a lethal dose. If we look lower in the table, we'll see aspirin and ibuprofen, which most of us have in our homes. And as you can see the difference between the amount of tablets that we can ingest, if we want a beneficial effect versus lethal effect when you consume 60 tablets or 105 in the case of aspirin. And you might think that that's not something that happens, and we hope it doesn't, but this will be the reference doses or the concentration to keep in mind. So we have discussed how substances move in the environment, how can we expose to them. So now the important part is that we're gonna be talking is once we are exposed to them, what happens to them in our body? And for that, the ADME process is what describes what happens from absorption, distribution, metabolism, and excretion. It's really the process that describes the behavior of the substance in the body. So the toxicity of a substance depends, of course, on the dose, and we saw that already in the dose response curve. However, how much chemical interacts with the target organs or systemic effects depends on the absorption, the distribution, the biotransformation or metabolism, and excretion of the substance or contaminant. So we talk about how are we exposed, but it's not exactly the same as what are the routes of entry to the body. When we talk about routes of entry, specifically in the environmental field, there's three: inhalation to respiratory system, ingestion to the digestive system, and also through dermal contact through the skin. If you're interested more in the pharmacology area, there's also other routes of entry to the body, such as injections, implants, eye drops, and suppositories. A nice way to simplify, and I'm saying simplify because it's really a complex process. A nice way to simplify what happens with a substance once it gets into the body is this illustration. Here you can see what would be the absorption phase, the distribution and metabolism phase, and at the end, the elimination. So when we're talking about the routes of absorption, as I mentioned, you will have ingestion to the GI tract, inhalation through the lungs, and dermal contact or absorption through the skin. Then it will come the distribution, what happens with the substance, how it starts moving around the body. And it will go through the different organs once it gets into the blood system or the lymph circulation. And it will interact with different organs all around the body, including fatty tissues and bones, until it's transformed. The transformation or the metabolism is a key process, very important in order for a substance to be eliminated. And we'll talk about that. And as you can imagine, we need to make these substances water soluble in order to be eliminated by urine or feces or even sweat. So the key aspect here will be to transform a lipid soluble compound into a polar or more water soluble compound so it could be eliminated. However, there's key aspect about how to get that substance absorbed. A key aspect here will be cell membranes. I'm not going to go into detail about how the substances can be transported into an organ through the cell membrane, but keep in mind that this is a barrier that will control at a certain point what goes in and what doesn't. Okay, so the solubility of how soluble these compounds or these contaminants can be in lipid will definitely be a factor to facilitate the entrance to the cell membrane. We can also have smaller molecules such as alcohol and water that can pass through to the membrane more easy. In the walls of the capillary, we'll see a different behavior because pores between the cells are larger than most drug molecules. And that will allow them to pass freely without lipid solubility being a big factor. It's always important, but in this case, it might be able to pass. Also, I need to mention the blood and brain barrier. This barrier provides protection to the brain, and it's beautiful the way it works. And we have also the placental barrier that needs to be there to protect the developmental human being, as well as other organisms. As I mentioned, in terms of absorption, the GI is one of the ways to absorb substance into the body. And the absorption can occur at any place along the entire GI tract. But the degree of absorption is site dependent. So the factors for absorption would be the type of cells at the specific site. So the absorption when the food or the substance is in the mouth, or as they go into the stomach, or later at the intestine, and then at the end at the rectum. The period that substance is in the GI tract, also how long it's retained in that phase. And also the pH of the stomach and the intestine will play a key role on what happened with that substance. You can imagine that in terms of the mouth, under normal conditions, xenobiotics are poorly absorbed because they don't remain a long time in the mouth. In the stomach, however, which it's high acidity, or with the low acidity, I should say, is a significant site for the absorption of weak organic acids. And of course, something that we hear frequently is the food. How much food we had in the stomach would also interfere with the absorption of the xenobiotic. The part of the GI where the most absorption occur is in the intestine. The greatest absorption of chemicals and nutrients will take place here, especially in the small intestine. Can you imagine? You can imagine the intestine has really a large surface, and it will remain longer there, and therefore it would facilitate the absorption. In terms of the colon and the rectum, there's really little absorption, but there's still some, and that we can relate to that because there's, in terms of medication, suppositories that are used for specific medication. The respiratory tract is also a way how substances can be absorbed into our system, and absorption can occur at any place within the upper respiratory tract, but it will depend, of course, on the physical form of that substance. If it's a solvent, if it's a gas phase, or if it's a particle. Also, there's specific regions, and some regions will have more absorptions than others. The most important site for absorption in the pulmonary region is the pulmonary region, and also the alveolar sac. Oxygen, carbon dioxide, and other gases can pass easily through these membranes. But if we're considering toxicity of inhaled materials will depend whether it's absorbed and how lipid soluble this substance is. Also, absorption can occur through skin. Skin is the largest organ in the body. It covers the entire body, and definitely it works as it should be for protection.
So, but in the skin section, you have to also understand something called as pilosebaceous unit. So what you can see here is a pilosebaceous unit, which aids in the absorption kinetics, and the absorption is primarily via that unit, and that's the way how you have Vicks when you apply on the skin, or maybe the hormones as a patch working out in that point. So I believe things are clear till here, beta. We will have the lecture and for another 10 minutes more, beta. Is it okay?
Yes, sir.
Hello guys. Okay, beta. Sure.
if your skin is healthy. A healthy skin will protect you. We still need to keep in mind that the skin is a complex, multi-layered tissue that although it serves as a barrier for most xenobiotics, some toxicants can cross and have entry through the skin. An example of those would be occupational solvents. For example, organophosphate pesticides have poisoned agricultural workers. And for that, it's clearly through skin absorption. Also, in terms of warfare agents, we have the effects on the neurological system. For example, sarin, which can produce fast death. It's really lethal. Specific doses, that's why it's used as a warrior agent. And also, as I mentioned, industrial solvents such as carbon tetrachloride can cause liver injury. And most of the time, it could be by inhalation, but it can also be through skin absorption. So once it's been absorbed, it's already into our system, what happens? Well, distribution of a chemical requires that the toxicant penetrates the membranes, as we already mentioned. And it will first penetrate the cells of the capillaries, and later it will move around different organs and tissues, okay? Some factors that will affect how it's distributed is the concentration gradient, the molecular weight of that contaminant or pollutant, how lipid soluble they are, and how polar they are. So once they start moving around our bodies, I don't know if you remember that at some point I mentioned that the key aspect of metabolism was to transform that compound that is more soluble in lipid so that it can be more soluble in water and be eliminated. So that process is called biotransformation. A biotransformation in order to convert a non-polar liposoluble compound into a more polar water-soluble. Where does that happen? That can happen in different organs of the body. However, it's mainly the liver is the organ responsible for the majority of drug metabolism. And it happens in the liver, but the ones that are really responsible for that is a group of enzymes, specifically a family of enzymes called cytochrome P450. You can imagine them as a big family, a lot of cousins, and some of them will interfere or be in charge of certain substances, and some others will metabolize other compounds. But besides the liver, we could see activity of those enzymes on kidney, GI, skin, and brain. When I talk about these enzymes, they're not all considered within the same category. They're divided in two groups. Phase 1 enzymes and phase 2 enzymes. And remember, the purpose of these enzymes is to make these compounds more water soluble. And here you can see these two groups, phase 1 enzymes and phase 2, the reactions that take place or that are in charge for phase 1 are hydrolysis, oxidation, and reduction. And what results from that is that the compound or the metabolite, keep in mind that that xenobiotic now will be transformed. So it will not be the same molecule that got into our body. It will be transformed and will generate a metabolite. So for those that when you read a study, be sure what is the parent compound, which is the original substance that got into our system, and the metabolite is that new species that result from the biotransformation. So when they interact with phase 1 enzymes, they will have some increase in hydrophilicity, but not a lot. When they encounter phase 2 enzymes, then the hydrophilicity is much higher. That will then result in a polar compound, or it will be added a polar group that will make the compound more polar soluble, and it will be then eliminated. It will facilitate the excretion. Of course, that will take us then into the process of excretion, which is the last part of the ADMET process. The excretion is the elimination of the xenobiotic and its metabolites. Routes to eliminate toxicants or their metabolites are mainly urine, feces, and exhaled air. However, there's also a few ways, such as sweat and sometimes maternal milk. This will be a summary of the process, how we integrate the ADMET process if we want to understand how certain contaminants can induce carcinogen. You can see here at the beginning of the diagram, we have the potential chemical carcinogen. If the substance is polar, and it will just be eliminated more easily. However, if it's not highly polar, then it has to be metabolized. And during that process, something that I perhaps did not mention was that those metabolites can be really reactive. They just don't transform and go straight to be eliminated. No, they transform and they also react. They can react with proteins, with DNA, with other organelles, with the mitochondria. And if that happens, you can have clearly toxicity. You can also have repair, especially DNA repair. And our system, our body is beautiful, and you can see that certain repairs happen when there's DNA damage. And eventually, if that's so, then those DNA adducts in this case could be eliminated. But when there's no repair and the damage is maintained, then cells can be transformed and eventually result in tumor or in cancer. So this is just an illustration of how the ADMET process can be applied to understand how chemicals can result in carcinogens. The last subject that I mentioned we were going to get into was the risk assessment. Risk assessment is really a method. It's a process so that we can integrate all the information generated by scientists and toxicologists to end with a recommendation or a characterization of what is the risk associated with that compound or with those pollutants. And this model, in this case, is as described by the National Academy of Science and National Research Council. And it's based for 4 steps: hazard identification, exposure assessment, effect and consequence assessment, and risk characterization. How do we use or generate the exposure assessment? Again, as I mentioned, we could have contaminants in the environment, but unless we monitor the presence and we know the level, we cannot be certain. So for us to determine the actual exposure of those substances in the environment, air, water, or soil, we need to monitor. We need to monitor the presence. And for that, there are multiple ways of monitoring the presence of contaminants. But also body, not only is in the air. In order for us to be sure that a subject has been exposed, we need to find something in the body that give identity to that exposure. And for that, we can have biomonitoring. For example, in the case of alcohol, when we are driving and somebody stopped to assure if there's alcohol in their system, you use exhale breath. So you can identify if there's the presence of organic solvents through that process. Also, lead in bone. If we have been exposed to lead for many years, you could find it in blood, but really in bone is where you would find it for many years. It will accumulate with time. Fatty tissues could also be used to measure the presence of fluorinated hydrocarbons. And also other ways of identifying the presence of contaminants or parents or metabolites in our bodies would be taking urine and blood samples. And for that, we apply analytical techniques to monitor the presence of those contaminants on the matrices. The third step in the risk assessment, as I mentioned, was the effect and consequence assessment. For that, there's many methods that can be applied, but you can see them in 3 categories: in vivo methods or in vitro methods. In vivo methods are pretty much methods using the entire organism. You'll use animals or fish, or maybe epidemiological studies or occupational studies, where we really seeing the effect of those contaminants or xenobiotic on the organisms intact. However, that's not always possible, and toxicologists also rely on in vitro methods. where we take cell cultures or cells from the organisms and we grow them in cultures under specific circumstances that will make them happy, so that they can behave not exactly as a body, but as close as possible. Another way nowadays, in silico models or computational analysis is also a way to understanding or trying to figure what are the consequences when we are exposed to those substances. Last, in the risk assessment method is the risk characterization. For here, you will integrate all the toxicological information, all the dose response curves, all the information that all those studies have generated, and integrate it in order to generate a consensus of what is the risk associated with that substance. For those that are not familiar with this field, I would recommend you to go to the ATSDR, the Agency for Toxic Substances and Disease Registry. It's really a portal that the CDC has generated. It's fantastic. Gives you a lot of information of a list of substances, not only a priority list, but also a list of substances. And you will be able to find most of the information that relates to toxicity, such as the asthma process.
So guys, this is the last part of the information where you could see there is www.atsdr.cdc.gov. That means it's the site of CDC Center for Drug Control, the government site in US, which will give you the idea about the list and the ATSDR toxic substances portal. So this was what was environmental toxicology, which we needed to understand to go before starting up with the toxicological studies pertaining to humans as understood in the phase 1, phase 2 and phase 3 of it. So that's what we have covered today. Is this things clear to you, beta? I know this was a very extempore lecture, and thanks for having sat for about from 11 to 1. That's a long time. But any questions for me, beta? Hello guys, any questions? No, okay. Things are clear, beta? Environmental is clear to you then?
Yes, sir.
Yes, sir.
Okay, beta. So I'll not bore you further. I'll plan up another lecture where I'll start with the phase 1, phase 2, phase 3 of the clinical trials perspective, taking forward from here. So for today, thank you so much for your attendance, guys. It was very good and nice that you all have been there. I could see Ketki, I could see Ashna, I could see Rupali, Rinku, Annapurna, Riddhima, Payal, Neha, Muskan. That's nice. Thank you so much, guys. Very good afternoon to all of you, and have a nice time. Take care. Goodbye and bye. Bye for now. Bye for now, madam. Okay. Bye bye, guys. I'll see you then in the next lecture. Thank you so much, guys.
Thank